Hi, I'm Lynn Lang, and this presentation is Cloud-Scale Genomics. I'm making this presentation for the purpose of educating people who might want to work with me in my consultancy. And so the focus is on what we do in the consultancy and how we support bioinformatics research. However, if you're just wanting to learn how my group works, you're welcome to watch this. This isn't just solely for people that would work with me. I'm making this really for that secondary purpose because there's more need in the industry than there are people working. So what are the challenges that my bioinformatics researchers bring to the table? First, they want to understand, when using the cloud, what they should be using, which cloud and which services. They also want to understand how to get efficient utilization, how to figure out what it will cost to use the services at the scale that they need, and then usability. Most of the researchers that I work with have very little experience with the cloud. So whether they're using tools that were already built by some of their partners in the bioinformatics industry that run on the cloud, or whether there are workflows that they've built on their laptops that they want to run at scale on the cloud, they want to understand how to use the cloud in a way that's effective and efficient and works for them, given their research budgets. And of course, the scale that we're talking about is humongous. So they want to understand how the cloud will impact how fast their analysis will run and how much data they can run on the cloud in a way that works for their budgets. So one of the things that I used a lot when I was teaching myself this domain, because I worked in other big data domains prior to coming into bioinformatics, is I used a lot of visuals. I also have had the advantage of being able to actually tour a facility that does sequencing at scale, actually multiple facilities. And so some of these photos are from those tours. So at the heart of it is the data gathering process, which happens in the lab, of course, because the samples, whether they're coming from humans or animals or something else, they're DNA and RNA samples, and they're processed on these very large machines. Speaking of DNA and RNA, more and more of my clients are having needs around what's called single-cell RNA workflows, and this could be in response to the effectiveness of medications, whether it's COVID medication or cancer medication. What's the impact in the body? So that is really just impacting the amount of data that they're needing to process. I still have clients that are working with DNA data sets, which are, of course, 3 billion letters. But single-cell RNA is more and more something that I'm seeing in my client base. And with all these slides, notice on the bottom right, I have a source linked, and this is some sort of explanation or video that has been helpful for me in understanding these basic processes so that I can help to speak the domain language when I'm working with my genomic researchers. This is an actual flow cell where the genetic material is placed by the researcher as it's processed in the various steps, which differ depending on what they're doing with the results. And this is what's put into the large machine for analysis to see what is the sequence in the particular section of DNA or RNA that they're working with. So I really liked this graphic. It's from a paper, and this is looking at the progression of disease. So this shows kind of from the researcher's mind how they think about collecting data. So they have a tissue or a tumor, they need to isolate and sequence individual cells, and then they need to look at genes, and those lines represent segments of DNA or RNA, so letters. And then read counts are from the cell, what they are able to get, and then the green and red is a visual tool, which is often used because of the amount of data. So they're comparing the gene expression profiles of single cells, and this was talking about single-cell RNA, to a reference to understand the tumor progression or the effectiveness of medication. So when I talk about data volumes, there's big data, huge data, and genomic data. So one of my clients, the Broad Institute of Harvard and MIT, this is published, and I have the link on the slide here. They are putting, on average, 45 terabytes per day into the Google Cloud. It's their preferred cloud, which is just an unheard of data volume. So one of the big challenges in supporting researchers is understanding and figuring out how to effectively work with the data volumes that are being generated. This is per day, and there's a link to the source. So as we drill in, there's our sequencer machine. And again, this is from a vendor, Illumina, which I've watched a lot of their videos to understand in more detail how sequencing actually works physically. And we start to drill into the steps of the data pipeline that we support when we work with our researchers. And in this particular case, we've got 3 different steps. And you can see that in each step, the genomic information, the NGS reads, is transformed, and then it's transformed in this case to 3 different file types. So it comes as a FASTQ, and then it's transformed to a SAM or BAM file, and then a VCF file. This is another aspect of working in this domain, is that the file types have a certain vernacular to them. We don't very often see CSV or text files, for example, other than metadata. The actual data itself is in these formats that are specific to genomics. Now, I do have some references on this with examples, because this is something that I know I struggled with because I wanted to understand what the underlying file type looked like. Sometimes they're binary, so to kind of, decompose them and so that they could be human readable so you can understand what you're working with is something that I've done the work to do, and I have some resources for you so you can kind of look at the files. I really like this illustration because this helps to understand the computational complexity of processing and analyzing DNA or RNA. This particular case is DNA, but you can see there's different methods here on the top: whole genome sequencing, whole exome or targeted sequencing, and whole genome or GWAS, of course, is the computationally more intensive because it is a broader coverage of the genome. And you can see on the bottom here that we have steps, and it starts with the clone DNA, and then it's broken into small segments, and this is really key. And those are unordered sequence segments. So you can think of them as slightly bigger pieces of a puzzle, and they have to be put back together. And notice on the bottom the resulting overlapping sequence segments. This is really a key in understanding the pipelining needs, that you don't have just 3 billion letters laid out in a row. You have these segments that have to be aligned and assembled. And because this is coming in from living material, there is potential for dirt and contamination and all kinds of things to go wrong. So there's many quality checks, depending on what the results will be used for in the pipelines that we support. In addition to supporting the pipelines themselves, another really important aspect of this domain is to have pipeline configurations that could be reproduced. When researchers get a significant result, they will often publish a paper, and more they are publishing the pipeline information so that their research can be reproduced. This is, in fact, one of the reasons that I came into this domain 6 years ago now when I started. There was a study done of, I think, 50 different published papers, and 75% of them were not reproducible because the pipeline information was not listed in a way that was reproducible, or the input data was not available. And coming from other big data domains, fintech and ad tech notably, I had familiarity with patterns in cloud for reproducible pipelines that I wanted to help to bring to the research community. Again, I think it's really important to understand computationally what's happening with the analysis of the data. So you can see here that we have in this case on the top, consensus sequences. So this is around some situation that you're studying. This could be some data set. It could be something with a genomic variant that has a disease condition. It could be, it really doesn't matter, but it's some sort of like baseline. And then the fragment reads on the bottom are what you're comparing it to. And you'll notice again how the fragment reads are different lengths and they have to be aligned. Notice, for example, in the CSV1, you've got an A and a C, and then you've got on the bottom A, G, A, C, C. So the way that these results are calculated is probabilistically. So in other words, how many C's do you have, how many A's do you have, how many G's in this case, and what is the most likely result? So everything is probabilistic, which is important in terms of the computational complexity because it's not a Boolean yes or no. You have this matrix effect in the pipelines because you are looking probabilistically at each of these locations. And again, this is just another view that I liked. I have all my sources linked so you can look if you're interested. But you can see how you have gaps. You have things that it's actually called, officially it's called junk DNA. And then you have sections that are of interest. It's interesting that it's some really small percent. I think it's 99% alike we are. I think that's right. It could be 97%. Anyway, it's over 90. And so the non-coding DNA is something that up until recently wasn't analyzed because of the cost of analysis. And there's actually been books written now about whether those sections are impactful in terms of activating different processes in the body. So it's interesting that now that we can sequence the whole genome or GWAS, in some cases, more and more researchers are. So just again, this is the kind of data that is running through the pipeline. This is from the Broad Institute, from one of their processes. And again, this just gets into a little bit more detail. So what the thing here is to generate a BAM file so you can find the variants of interest, the varying letters. And there's 7 steps here. So one of the things about the pipelines is they can have many, many, many steps. Depends on, again, if they're just doing rough research on something new, or if they're doing something that is going to apply to human health, there's going to be more and more quality checks. So you can see that in each step level, there's sub-steps. So for example, in the step 1, generate the UBAM, there's a FASTQ and an aligned BAM, and these steps run using processes, and then they will take the results and aggregate them into what's called an unmapped BAM file. So again, it's not so important you understand each of the processes. It's more important that you understand that the processes are standard and they are used depending on what is the need of the researcher. So in this case, they need to generate a UBAM, mark adapters and align with BWA, and then merge for raw BAM. That's what they're needing to do here for variant discovery. So in terms of the pipeline, you see you've got the blue boxes, 1, 2, 3, 4, 5, 6, 7, 8, 9 different processes that you have to focus on in terms of optimization. And that's really where me and my consultants come in to figure out how to make those steps run faster, or tasks as they're called. Now that was an extremely fast introduction to bioinformatic pipeline processes. My experience, of course, is in application of high-performance data pipelines on the cloud. And so what I work to do is to take that knowledge and apply it to those types of problems using patterns. The first step is to figure out how to use the cloud and which cloud to use. You might be surprised to know that for many bioinformatics researchers, they haven't had the opportunity to work with the cloud, even at university. So when they get their first postdoc research role with a research organization like the Broad, they are often completely new to working with the cloud, which is one of the things that we try to address. Now, other folks have had some experience, but usually it's not a core focus. Although there are other clouds out there, these are the ones that I focus on with my clients. So Google Cloud, Amazon, or Microsoft. So as I have been teaching clients, bioinformatics or not, about using the cloud, I have 25 courses on LinkedIn Learning, and I'm making companion repositories on GitHub for all of them. Now I have generic courses, like you can see near the bottom here, GCP Learning Cloud, I should say here in the middle, which is a index repository that has resources for learning cloud in general. But you can see near the bottom, I have GCP for bioinformatics. This is a course that I made, and it's on GitHub only. And it's for bioinformaticians, and the idea is that they can get the basics of GCP using bioinformatic data and bioinformatic processes. So, one of the takeaways from watching this talk, hopefully, is that you will look at some of my repositories because I have a lot of information there. So the general pattern of moving to the cloud at scale is this pattern called a data lake. Now, it's as simple as possible way to look at it. You have no database. You have buckets because you use files as your data. And like we were looking at, they could be BAM files or VCF files or whatever, but they're files. And you have input files, and then you have data lake, and then output files. And then you have compute processes that act on them. And those are those processes that we saw that are required in the pipelines. And they are expressed as tasks. Now, this is just kind of to get us thinking about this pattern. In the existing research that I see, oftentimes people are running things on their laptops. And so they're running at very small scale, and many of the research organizations make minimal use of the cloud. They often have an on-premise high-performance compute cluster that the researchers will have to connect to, they'll have to SSH to, and send their files up to whatever workflow as a batch job, and then wait for it to complete. So they're completely isolated from what's actually happening. And so it's a big change to be working with the compute itself. Usually, like I said, researchers are either working on their laptop or they're using HPC. If we overlay this pattern into genomic data analysis, you still have the input and the output. We have the tasks, but oftentimes there'll be intermediate files that are saved for, they're needed for the tools, or they're needed for compliance. And then take note of tool number 3 here, which is scattered. This is a typical pattern. It's called MapReduce or scatter gather, and the idea is you have some computationally intensive task in your pipeline, and you need to scale out. If you are using HPC cluster, then you're using compute resources there, and you just basically have to wait. Modern cloud pipelining allows for burstable compute. The most cloud-native way to do this is serverlessly with functions, but most researchers are not doing that quite yet. They're working with Docker containers or virtual machines, but usually Docker containers now, which are subsets of virtual machines, if you will. So the pattern in general on the cloud, and this one is for AWS, is you've got this data lake where the files live. And then you have a set of services, and let's start with the bottom. You have virtual machines that make up a cluster. Now what I find when I work with researchers, most of the researchers, if they use the cloud at all, they simply configure and spin up virtual machines. And they often are really frustrated because they don't have the background in it, so it takes them a long time to do it. So that's a cost in terms of the time. It also is expensive because they turn them on and they don't want to turn them off. They kind of think of it like an HPC cluster on-prem, and that's sort of the antithesis of cloud native, which is turn something on, use it while you need it, and turn it off, kind of like electricity or water in your house, because it's billed by the time interval. But again, that pattern is not very well understood because it's just not something in the wheelhouse of most researchers. Now you can do that manually, but within the cloud services, there are higher level services. In Amazon, it happens to be something called Batch service that will orchestrate virtual machines turning on and turning off for you. So some of the higher level tools that are being created by the research organizations, and I have in purple here a tool called Cromwell, which is physically a Java jar file that is created by the Broad Institute. This is an orchestrator that runs on top of AWS Batch, and the idea is that it abstracts the orchestration to a level that is more understandable to a researcher. There is a language that comes with Cromwell that's called WDL, which is shown higher up here with the pipeline. It stands for Workflow Definition Language. And the idea is that you write the pipeline the way you would think of it as a researcher. So you write a workflow name, and then you write task names. And the tasks are then associated with compute, which would be VMs or Docker containers, that Cromwell then talks to Batch and spins them up and spins them down only when they are needed so that you have cloud efficiency. So this is a lot for researchers to work with. And one of the areas that my consultancy does a lot of work is researchers will hear about this because they'll overspend basically their compute budget if they just turn their VMs on because it's not efficient, costs too much. They don't need to be on all the time. And they'll want to implement this pattern, but you can probably tell just from looking at the diagram, this is rather complex to implement on your own. Now, I have worked with some teams who that is the best fit for them, to help them to design an architecture like this for their particular research. That usually is teams that has a dedicated IT team as well, a DevOps team, and not all research teams have that. Because of that, there are some pipeline tools that are created at a higher level that I do quite a lot of work with as well. So the ones that I've worked with most recently are on the left in green. It's Terra.bio, and this is looks like a website. Basically, it's a graphical user interface that allows users to run WDL workflows using the Cromwell orchestrator on Google Cloud currently, although they have announced and they are working with Microsoft to run on Azure as well. So the underlying orchestrator for Google Cloud is life sciences, so Cromwell works with life sciences, and for Azure, it's Azure Batch. On the right, we have Nextflow Tower. This is in some ways competing. This is put out by a group called Secure Labs, and this is a graphical user interface that runs workflows based on the NF language, which is similar but not the same as the WDL language. And again, it is designed to be at the level that the researchers think can work, and then it integrates with AWS Batch, Azure Batch, or Google Life Sciences. So these tools have proved to be very, very popular, as you might expect, because in the community, they're much more usable. That being said, they're relatively new, and so some work that my group does is around adoption and usability and understanding how to use these tools, because although Terra was developed at the Broad, there are many, many Broadies or Broad researchers, and within the Broad itself, there's a training effort to get everybody working on this, and there's also an effort to work with partners. Nextflow has a commercial tier. It has a free tier and a commercial tier. And again, as they've been developing, they have had very rapid growth, and I've had customers who I've worked with to learn how to use Nextflow as well, Nextflow Tower as well. So when the researchers are trying to figure out how to use the cloud, they have basically 5 different levels. Now, a lot of times they want to start at the bottom, and so because that is really where they think of their research, like they create the task, the analysis task, and they write it in C++, Python, or R. But more and more, you want to start at the top, because as these tools start to become standard, Terra.bio, Nextflow Tower, and there's another one called Galaxy Project, and there are more. These are research. There are also commercial versions. This is emerging as the method of getting your data up into the cloud and being able to analyze it more effectively without having to do all this underlying work. It is kind of a vertical in progress, though. I would say about maybe half, maybe 2/3 of my customers are working at the use workflow level, but there's still a good bit of them that want to work at a little bit lower level. Now, if you use a workflow, then you need to understand like what's available, which workflows are there, and then how to configure them. So configure the input and output, and then very importantly, the workflow parameters, like how much compute do you want to use, and what's your batch sizes, and many, many, many parameters, so that you can run your workflow within your cost structure and your time result. I've worked with quite a few customers who are trying to learn Whittle. In fact, I created a course for the Broad called Learn Whittle. And I've also worked with customers who are trying to learn Nextflow. JSON is the underlying workflow language of Galaxy. Galaxy is more of a visual tool. I have not worked with customers that want to write JSON. They usually use the Galaxy Visual Workflow Designer. Underneath that, there are tools that are standard, the most notable of which is the GATK, which is the Genomic Analysis Toolkit. It's a set of tools from the Broad. Again, it's a jar file, has literally hundreds of tools in it. That is often used as part of WDL workflows on Terra. So it works together, it's designed to work together, and it's important because it's optimized. There are other open source tools, kind of one of the ones that's been out there for a really long time, something called Blast. There's bed tools, there's literally hundreds of tools, which is kind of a challenge in the industry because the way that it kind of started is everybody just wrote their own tool in their own university and didn't optimize it for scale. So GATK in particular is trying to address that. Now, if I had a customer that wanted to build their own analysis, did not want to use Terra or did not want to use Nextflow Tower, then what would that look like? And this is an example from Amazon. So you can see in the left side here, you've got the sequencer and the files are going into the Amazon file storage S3. And then you've got clusters of compute, that's the orange. So those are EC2 instances. And they've got a lot of detail here. You can see they've got the size of it on here. And notice it's managed by AWS Batch, the first one, host filtering and quality control. And then you have another group of tasks. And again, this is another important aspect of the work that we do. We help to analyze, do we need one set of compute for one task because of the amount of work it is, or do we need to group the tasks together? And these are the subtleties that can really make a big difference in terms of the time and the cost. And you can see that the EC2 auto-scaling group 2 here is doing assembly-based alignment, and there's a nice graphic on the bottom which is showing you the reads in blue, red, black, and green. They're showing using Blast, that tool I talked about, and then the assembly-based alignment with the different reads that are being matched to the contigs. And then in this case, we're looking for different species. So, and then we're doing taxonomic reporting. And that's the third step here. And then we're outputting to a database and visualizing. So again, not really important you know all the details. It's just kind of important you understand the difference in work, basically, whether it's configure a Terra workflow or whether it's build a workflow from scratch and optimize it, because those are substantially different. And my consultancy gets involved in both. And this is just to give you kind of a visual. If you were to use a workflow language on the right here, and I've just collapsed it so that you can see how the overall sections, the green, blue, and purple, correspond to individual tasks. And this is on the right, it's WDL. I collapsed it down so you didn't see any of the detail, but each one of the calls is calling a specific tool out of GATK. So strangely, surprisingly maybe, one of the things that I've worked on in addition to optimizing and building is discovery, because this is a lot of work to go from running on your laptop or HPC to the cloud. So if you've got something out there that already works, grabbing it and running it on a cloud is a great way to get moving. So in the case of Terra, there is a showcase for WDL workflows where there's a wiki and oftentimes there's a diagram and sample data. There's also another website called DocStore that is a home for WDL workflows, for Nextflow workflows. It's a registry, basically, and it includes a visualizer. And then the Broad has what's a section called the methods repository as well. So the pattern is that there are registries of workflows. There's also registries of containers, or some registry called biocontainers, because it is important to reuse tools and tasks that are optimized rather than build everything from scratch. So again, the general pattern when running analysis jobs is you have some sort of dev environment, local laptop, cloud, or HPC. Another aspect that is often challenging is data. When people are moving to the cloud, oftentimes they only run local or HPC, so they don't have a small data set. And sometimes they also don't have an anonymized data set. A lot of this is human health, and so there's HIPAA rules around it. So you have to have some data that can be worked with if you are going to partner with someone like my company, because I don't have a researcher credential. Also, it's interesting that there is more and more data being put up into the cloud as repositories. This was particularly accelerated in 2020 and 2021 because of COVID. And it is becoming more and more of a trend to make data available in the cloud, which is actually great because you don't have to download it. You can just access it from the cloud. If you're using WDL, then of course you're going to need to either write a workflow or find one. You're going to need to figure out how that integrates with the tasks. You're going to need to make a JSON file which shows where your input files are coming from. And then you're going to need to configure your workflow. So it's not as simple as just grabbing it and saying input and output. The configuration of a WDL file requires some understanding of what you're doing as well. So oftentimes what my clients want me to do when we go beyond moving to cloud is pushing limits. So it's a very common task that I'll have clients that have written something in one of the big languages, WDL or Nextflow, and they want to translate it. There is no good tool, and so that's something that I've done for some clients because they just have different needs. They want to collaborate with different research groups that are using different tools. Maybe there will be a tool, it'd be great, but we've done the work pretty manually. Genomic analysis tasks at scale and speed. There is a version of GATK that runs on a super efficient processor. It's like a super CPU. It's called an FPGA, and it runs 10 to 100 times faster. So we did some performance validation for clinical diagnostics to prove that paying for FPGA on the cloud would result in the performance improvement that was needed. So for example, you could go in for a doctor and you could have an analysis done and get the results back very, very rapidly so that your treatment plan could be altered depending on your results. Compute controllers and storage. The computational complexity of analysis often calls for machine learning, sometimes neural networks with TensorFlow. And on Google Cloud, you have GPUs and TPUs, which are super GPUs. They cost more, but again, it's this time and money thing. Another thing that my group has done is support performance testing cloud storage services, again, for speed. And in the compute cluster area, using AWS, Azure, GCP Batch services to dynamically provision cloud and containers, really understanding how those higher level languages, whether it's Cromwell from the Broad or whether it's Nextflow, integrate with the different services. You know, it's tricky because Cromwell and Nextflow are, of course, evolve. So do AWS Batch, Azure Batch, and GCP Life Sciences or Batch. And so keeping those things in sync requires work. So I have been working for, gosh, I think it's 5-6 years now, trying to educate myself on bioinformatics and bioinformatic research needs so that I can support using cloud pipeline patterns, helping the research go faster and be cheaper. And I have the results of that out on this GitHub repo, Team Terry. In case you're wondering, Terry is the reason I got into this. She had cancer 6 years ago and was not able to get into a clinical trial because it wasn't available and had a very almost inhumane course of chemotherapy and was, nearly died. But she didn't. She recovered. However, to this day, she has to have treatment, and I just feel like for all the people out there like Terry, we need to use our full power of what we can build on the cloud to help our researchers get better results faster. It's all about impacting health. Another interesting thing, in 2020, one of my clients was working with integration with this visualization tool to understand COVID variants. And it was quite startling as I was doing this contract because I got long haul COVID in 2020 and I'm living in Minnesota. So this is actually the variant, this is about the time I got COVID that I had. So it's kind of startling when you are working on a project and it impacts your own health. Not that I'm the only person of concern here, but we all deserve to have our information used in the best possible way. And again, by building together and working with our researchers, we can make an impact. So join me, I'm Lynn Langett, Bioinformatics Cloud. Thanks for listening in.